Assalamualaikum. So, my name is Rahman and I'm a first year medical student in UMS. Today, I would like to present about neurology. So, what is neurology? Neurology is a branch of medicine that focuses on the nervous system, including the brain, spinal cord, nerves, and muscles. It studies how these parts control movement, sensation, thinking, memory, and emotions. Neurologists diagnose and treat conditions such as headaches and migraines, epilepsy, stroke, multiple sclerosis, Parkinson's disease, nerve injuries, and problems with memory or thinking. Overall, neurology helps doctors understand how the nervous system works and what happens when it does not function properly. So, next thing I'm going to talk about is the blood-brain barrier. So, what is blood-brain barrier? Blood-brain barrier, also known as BBB, is a highly specialized selective barrier that separates the circulating blood from the brain's extracellular fluid, protecting the central nervous system while still allowing essential substances to pass through. Its main function is to maintain a stable chemical environment for neurons, which is crucial because even small changes in ion levels or exposure to toxins can disrupt normal brain function. Unlike most tissues, the brain cannot tolerate wide fluctuations in blood composition, so the BBB plays a critical protective and regulatory role. Structurally, the BBB is formed primarily by endothelial cells lining the brain's capillaries. These endothelial cells are unique because they are tightly sealed together by tight junctions composed of proteins such as claudins, occludins, and junctional adhesion molecules. These tight junctions eliminate gaps between cells and prevent substances from slipping through the space between them, forcing most molecules to pass directly through the cells instead. This is a key biochemical feature that distinguishes brain capillaries from those in other organs. In addition to endothelial cells, the BBB is supported by several other components that together form the neurovascular unit. Astrocyte, a type of glial cell, extend their end feet to surround the brain capillaries and release signaling molecules that help maintain tight junctions and BBB integrity. Parasites, embedded in the capillary basement membrane, regulate blood flow, vessel stability, and immune cell entry. The basement membrane, made of extracellular matrix proteins such as collagen, laminin, provides structural support and further contributes to selective permeability. Next. Functionally, the BBB is highly selective in what it allows to cross. Small, nonpolar, lipid-soluble molecules such as oxygen, carbon dioxide, and some anesthetics can diffuse freely through the endothelial cell membranes. Essential nutrients, including glucose and certain amino acids, are transported across via specific carrier proteins like GLUT1 for glucose. Ions and water are tightly regulated by specialized channels and transported to maintain proper electrical activity in neurons. In contrast, large molecules, toxins, pathogens, and many drugs are blocked from entering the brain, which protects neural tissue but also make treating brain disease more challenging. The BBB also plays an important role in immune regulation. Under normal conditions, it restricts the entry of immune cells and antibodies into the brain, helping to prevent excessive inflammation. However, during infection, injury, or inflammation, the BBB can become more permeable. While this increased permeability may allow immune cells to fight disease, it can also lead to brain swelling, edema, and neuronal damage if not properly controlled. Clinically, dysfunction of the blood-brain barrier is associated with many neurological disorders. In diseases such as multiple sclerosis, BBB breakdowns allow immune cells to enter the CNS and attack myelin. In stroke and traumatic brain injury, BBB disruption contributes to inflammation and tissue damage. In neurodegenerative diseases like Alzheimer's disease, subtle BBB impairments may interfere with waste removal and nutrient delivery. Thus, the BBB is not just a passive wall but a dynamic, biochemically active interface that is essential for both brain protection and normal neural function. Next, I'm going to talk about the Parkinson's disease. So, what is actually Parkinson's disease? Parkinson's disease is a chronic progressive neurodegenerative disorder that primarily affects movement. It occurs due to the degenerative of dopamine producing neurons in a specific region of the brain, known as the substantia nigra. The The loss of the dopamine leads to impaired communication within motor pathways, resulting in characteristic motor symptoms. So, epidemiology of the Parkinson's disease is that Parkinson's disease is most commonly seen in older adults, with its incidence increasing significantly with advancing age. It's slightly more prevalent in males than in females. Although it is typically considered a disease of the elderly, early onset Parkinson's disease can occur, though it is relatively rare. The clinical features of Parkinson's disease is that patients with Parkinson's disease commonly present with a resting tremor, muscle rigidity, and bradykinesia, which refers to the slowness of movement. Postural instability may develop as the disease progresses. Additional features include a mask-like facial expression, shuffling gait, reduced arm swing, and difficulty initiating movements. Next, this is where the interesting part is, which is the pathophysiology of the Parkinson's disease. So, Parkinson's disease is mainly caused by the gradual loss of dopamine-producing nerve cells in a part of the brain called the substantia nigra, which is important for controlling movement. Dopamine is a chemical messenger that helps the basal ganglia regulate smooth and coordinated voluntary movements. So, as these dopamine producing cells die, dopamine levels in the striatum decrease. Dopamine normally helps balance movement controlling pathways in the brain. When dopamine is reduced, this balance is lost, leading to increased inhibition of movement signals. As a result, the motor cortex is less stimulated, causing slow movement, bradykinesia, and difficulties starting movements. Parkinson's disease also involves an imbalance between dopamine and another neurotransmitter called acetylcholine. With less dopamine, acetylcholine activity becomes relatively higher, which contributes to muscle stiffness and tremors. A characteristic feature of Parkinson's disease is the presence of Lewy bodies inside nerve cells. These are abnormal protein clumps made mainly of alpha-synuclein. Lewy bodies disrupt normal cell function and contribute to nerve cell death. In addition, problems with mitochondria and increased oxidative stress damage nerve cells. Dopamine-producing neurons are especially vulnerable to this type of damage, which leads to their gradual degeneration. Overall, Parkinson's disease results from dopamine loss, disrupted basal ganglia function, neurotransmitter imbalance, and ongoing nerve cell degeneration. Together, these processes cause the movement problems that worsen over time in people with the disease. Next, I'm going to talk about the neurologists that contribute a lot in this type, type of medical field. Santiago Ramon Cajal, born on May 1, 1852, in Patilla de Aragon, Spain, is widely regarded as the father of modern neuroscience. Despite struggling academically in his early years, he developed a strong interest in biology and a remarkable talent for drawing, which would later prove crucial in his scientific career. Cajal pursued medicine and eventually became a professor of anatomy and histology and eventually became a professor of anatomy and histology at the University of Barcelona and later at Madrid. In 1906, he was awarded the Nobel Prize in Physiology or Medicine, an honor he shared with Camillo Golgi, in recognition of their pioneering work on the structure of nervous system. Cajal's most significant contribution was the neuron doctrine, which fundamentally changed our understanding of the nervous system. Uh, in the neuron doctrine, before Cajal, many believed in a reticulatory theory, where the nervous system was a continuous network. Cajal used Golgi's staining method to show that neurons are discrete cells that communicate via specialized junctions, later called synapses. Next major contribution of him is that neuroanatomical discoveries, which he provided detailed illustrations of different types of neuron, including Purkinje cells, pyramidal cells, and granule cells. He also studied the structure of the retina, cerebellum, hippocampus, and cerebral cortex. His drawing remains a reference in your anatomy textbook. Next is brain plasticity, where he suggested that neurons could form new connections, laying the foundation for understanding learning and memory. He also pioneered ideas about developmental neurobiology, including axon growth and guidance. Next, he also contributed a lot in research philosophy, whereas he combined artistic skill and scientific observation. He also advocated meticulous observation and drawing as a tool to understand complex structure. So this is a few of Santiago's drawing. As you can see, he draws a lot, and that's all from me. Thank you.